We're already on our fourth day of this genetics unit, and today we're going to tackle mitosis. So by the end of today, I want you to be able to describe what happens to chromosomes at each different stage of mitosis. Also, I want you to know why organisms do mitosis, both multicellular organisms, organisms that have more than one cell, and unicellular organisms, which are organisms that only have one cell. I want you to be able to compare and contrast a parent cell, which is what we call a cell before mitosis, to its daughter cells, which is what we call the resulting cells after mitosis. Finally, to tie this into GMOs, I want you to think about and be able to explain what's going to happen to a gene that we put into a GMO after it undergoes mitosis. Will that gene go into the daughter cells, or will only a certain percentage of those daughter cells have the gene put in there? Let's jump right in. We've got 5 more slides today. Be sure to follow along with your notes and pause or re-watch parts of this video as you need to make sure that you catch all of the notes. So this first part, we want to think about, why do cells divide? And sometimes mitosis and cell division are used interchangeably. So it's okay if when we talk about mitosis, we call it cell division, or when we talk about cell division, we call it mitosis, at least for now. So why do cells do this? Well, it depends what type of organism the cell is in. For multicellular organisms, organisms with more than one cell like us, we use mitosis to grow and mitosis to repair our tissues or regenerate our tissues. So here's an example of how we grow. We all first start as a sperm and an egg, and we get bigger and bigger and bigger. And our cells actually stay pretty much the same size, and how we get bigger is by having more cells from cell division. Another thing we have to do is to repair our tissues when we get wounded, just like this person that got a cut on their finger. So what happens, first, there's a blood clot that stabilizes that wound so it stops bleeding, and then as the wound heals, it heals as a result of cells starting to divide and fill in the gap that's left from the cut. Now if you're a unicellular organism or a one-celled organism, mitosis means a whole different thing. So a really good example of a unicellular organism is a bacteria. Just like the bacteria we were talking about yesterday, E. coli. And yesterday we reprogrammed E. coli to make RBST to be able to inject into cows and make more milk. So why would something like E. coli do mitosis? Well, it can't grow or repair anything because it's only made up of one cell. So when E. coli divides, it's kind of like it's having babies or it's reproducing. So this type of reproduction is called asexual reproduction. And the reason it's called asexual reproduction is because bacteria don't have to have sex to reproduce. Instead, there's only one organism or one parent, and the children or offspring cells are genetically identical to the parent. So they're kind of like clones. So here's a picture showing that process of asexual reproduction or mitosis in bacteria. You can see that you start with a parent cell and you end up with 2 offspring or 2 daughter cells that are genetically identical to the parent. Let's learn a little bit more. If we zoomed in and take a look at what's happening to cells during mitosis, you can kind of narrow things down into a couple of basic processes or basic events. The first thing that needs to happen, since we're making copies of cells, we need to make copies of DNA. So very early on in this process, in interphase, the DNA is copied. Then the DNA is going to be shuffled around so that each daughter cell gets the same exact amount and same exact copies of DNA. So you need to make sure that one copy goes into each cell. And that's going to involve a lot of organization and sorting. So, the second basic step that happens for mitosis is that the chromosomes condense or get packed up so that they can be sorted and moved around really easily. And this is just the same thing like you'd do if you were moving your house. You'd put everything in boxes and pack it up so that it would be easier to move around. After the chromosomes are condensed, then what happens is they're moved. First, they're moved to the center of the cell to check, make sure they're all there, and they buddy up. And then what happens is the chromosomes are split and go to opposite ends of the cell. And these parts of a chromosome, like a chromosome at this point, is shaped like an X, and each half of the X is called a chromatid. And because they're identical, they're called sisters. So each sister chromatid goes to opposite ends of the cell during these two processes, anaphase and telophase. Once that's done, the cell divides and the chromosomes can unpack and become diffuse. So one thing that I really want to make sure you understand, and I want to stress, is that the end result is that the daughter cells at the end of mitosis are the same as the parent cells that started this process. So they're genetically the same. They have the same DNA since it was copied very early on. Now, a word I've been throwing around a lot is chromosomes, and I want to make sure that you really understand what a chromosome is. So a chromosome is just kind of a way that DNA is organized and DNA is packaged. And we've been talking about genes and how you can take one gene out of one organism and put it in another organism to make a genetically modified organism. So how each chromosome is organized is that it's a whole bunch of genes. And what a gene is, the definition for a gene, is a sequence of DNA or a stretch of DNA that codes for a trait. So, for example, remember, bacteria make that Bt toxin that's able to kill the corn borer and other insects. Scientists took that gene out of bacteria and they were able to put it in the chromosome of corn so that the corn could make that same toxin and kill corn borers. So each gene is a piece of DNA responsible for a specific trait. Now, the way that chromosomes tie into mitosis is that chromosomes are condensed or packed up during the phase of mitosis called prophase, and they do that so they can be easily moved and sorted during the rest of mitosis, metaphase, anaphase, and telophase. But when they're doing their normal job, like in the cell cycle game during interphase, the chromosomes spread out and relax, and when they're in that phase, they're called diffuse. The reason that they do that is so that all of those genes in there can be accessed and used. So what I want us to be thinking about is if you go through all that trouble and insert a gene like RBST into a cell like an E. coli cell, when the E. coli make baby E. coli or daughter cells, will that gene that we put in there still be there? Will it only be there in some of the cells, or all of the cells, or just the cell that we started with? What I want us to do now is to model mitosis using pop beads to answer this question. Let's do it.